Hey guys, Kenna here. So we're going to go ahead and go over some essential questions on DNA replication. But this is kind of a key piece. It's similar in some respects to what we did with protein synthesis, except we're not going to go out and make proteins. We're simply making a copy of the original DNA molecule for when cell division happens in mitosis, so that each daughter cell ends up with an exact copy of the original DNA sequence. Okay, so what are our essential questions? What is produced in DNA replication? And I just kind of answered that, but we'll go into more details. Two, how does the process of DNA replication make sense with respect to both the stability and mutability of species? And three, what is an Okazaki fragment and why is it needed? So, first of all, when I say replication, hopefully the word sounds familiar to you. It simply means that we're duplicating, we're making a copy of the DNA. If you remember back when we were talking about mitosis, this happens during the S phase of interphase. This is the DNA replication portion. And it has to happen before we do mitosis or meiosis, which we'll go ahead and talk about on Thursday, because we need to go ahead and have enough DNA to go into each one of the daughter cells. Our first step, as we talked about, is part of the double helix has to be unwound. We can't do all of it at once, if you remember, because the DNA that's in a single cell, if it all stacked end to end, is over 2 meters long. That's way too big. So we just unwind little sections at a time, make our copies, and wind it back up as we go. But because the DNA double helix has two strands going in opposite directions, it can't just copy straight through on both strands. Replication is continuous on one strand, the leading strand, and in small pieces called Okazaki fragments on the other, what's known as the lagging strand. These little fragments of Okazaki fragments are then stitched together later on to make a continuous piece of DNA. So remember, when we're going ahead and doing replication, we're going to go ahead and make a copy of the DNA molecule so that we have two sets, so that when we go through the division of mitosis, the two daughter cells at the end of telophase and cytokinesis both have the same amount of DNA as the original cell. The key proteins that we see that help with the winding, unwinding process are these proteins over here, either nucleosomes that we're going to help with it, or there's another type of enzyme that we'll talk about a little later that has to be utilized as well. Okay, so when we start taking a look at this, here's your mitosis wheel. You've got G1 or gap1 phase, which is going to be where the cell grows. You've got S phase, where DNA replication takes place. That's what we're going to focus on today. You've got G2, where we go ahead and prep for mitosis. And then you've got the tiny little M phase, where the actual division occurs of the nucleus, and then cytokinesis, which separates the cytoplasm to produce our two new daughter cells. All right, so let's go over this process. You will see some things that are in common with transcription in particular that we talked about last term. Before a cell divides, the DNA strands unwind and separate. Remember, this is why the bases are connected with hydrogen bonds, so that the energy requirements to do this part are not incredibly high. Each strand then is going to make a new partner strand by adding the appropriate nucleotides. These are going to bond according to Chargaff's rule. Remember, Chargaff's rule is A to T, C to G, adenine to thymine, cytosine to guanine. This way, each pair is matched up based on having a purine, which is a double ring base, and a pyrimidine, which is a single ring base. They'll be bound together by hydrogen bonds, and one set has 2 hydrogen bonds, the other set has 3 hydrogen bonds. When we go through this whole process, the result is there are now 2 double-stranded DNA molecules inside the nucleus. This is important so that when the cell divides, each nucleus of the daughter cells contains identical DNA, and this process is known as replication. Now, originally, when people were unsure about how the process occurred, they originally saw that DNA was probably going to be copied to produce one new strand, and then the other side was going to be the two originals. And what they've discovered since is that you end up having what's known as semiconservative replication. where each daughter set of DNA has one new strand and one original strand. This way, they can go ahead and base up and base pair up. And so when we make our copies of complementary pairing, you end up with one original and one new in each of the daughter cells. The DNA piece that is old is known as the template strand, and the new DNA strand is what's known as the complementary strand. So the old or original idea was the conservative model, which you see on the right here. And the current understanding is what's known as the semi-conservative model, which is here on the left. DNA replication takes place in the nucleus, which should again make sense. Just like in transcription, it has to take place there because the DNA does not leave the nucleus to keep it protected. The replication process is completed by an enzyme. Remember, in transcription, we talked about RNA polymerase. Here, we're talking about DNA polymerase, and it's going to copy DNA into DNA according to Chargaff's rule, which again is A to T, C to G. Here's a diagram of that process. And what I want to really point out here is that you've got DNA polymerase going in both directions. Remember, these two strands are facing opposite ways, and because of that, your DNA polymerase has to read in one direction. Just like when you read a book from left to right, if it's in English, and top to bottom, again for English, then it makes more sense if you go in order. And so the DNA polymerase reads it that way. And so as it goes through, you'll notice that we have what's called a replication fork. This is the part, or Y, where the DNA double-stranded portion is unwound and unzipped so that we can go ahead and make our copy. You end up with one original strand and one new strand in what's known as semi-conservative replication. Okay, there it is, semi-conservative replication. Now we run into an issue with that having to read in one direction. The fact that one strand goes one way and the other goes the opposite direction means that we can't necessarily read both strands straight through. So what happens? This is where Okazaki fragments comes in. These are relatively short pieces of DNA with a little RNA primer, don't need to worry about that so much, on the lagging strand of DNA. So if you look at the model on the right, you'll notice that one strand is going from 3' at the top to 5' at the bottom, and that the other strand goes from 3' at the bottom to 5' at the top. Well, this is simply a chemical designation for direction, okay? And the DNA polymerase is only going to be able to read from 3' to 5', so in sequence. Now, this was originally discovered in 1968 by two brothers, Riji and Suniko Okazaki. And what they discovered is that while that leading strand that is organized from 3' to 5' codes straight through without stopping, it has no problems, the opposite strand, the lagging strand, can't do that because it's organized from 5' to 3'. And so what it does is it waits a little bit, connects to the strand, codes from 3 to 5, the normal direction, and then waits a little bit and does the same thing, leaving behind these little broken pieces called Okazaki fragments. So this discontinuous nature of the copying of Okazaki fragments creates these problems in making it a continuous sequence because both of your DNA sequences have to be intact. And so what happens is another enzyme, another protein comes in and attaches those little fragments back together so that you have two complete strands of double-stranded DNA wound up in its helix, okay? So let's go ahead and take at that process. I mean, it's a lot to kind of digest right there. So let's take a closer look at that process and see how it goes. So if you remember, we'll bring back our little friend Dino DNA, and he goes ahead and creates out our double helix. First thing we do is unwind and unzip so that we can actually access the nucleotide bases. Once we've done that, each partner strand is going to build up by matching a complementary nucleotide to make two strands in terms of our complementary base pairing. So we're going to go through and we're going to match up base pair by base pair by base pair, and it's going to go in one direction. Remember, it reads from 3' to 5', and lays down from 5 prime to 3 prime, okay? And on the other strand, excuse me, you'll notice that we're going to go in the opposite direction. If you take a look at the point of the sugar, the 5-carbon sugar, on the left side, it's pointing upward, but on the right side, it's pointing downward. So in terms of that 3 prime 5 prime orientation, we have to remember that it's flipped. So what it does is it goes in and it puts the pieces in order, again reading from 3 prime to 5 prime, but it does it in little chunks. We didn't do the whole thing straight through. It waits a little bit, and then it comes through and does the same thing again. Then you're going to go ahead and stitch together this phosphate so that you have a continuous sugar phosphate backbone, and this one looks exactly the same as the one on the left. Okay, so that's essentially how DNA replication occurs. Now I have some little interactives to help reinforce that, because while it's not a whole lot, it's kind of complicated, and we want to make sure that you understand how this works. This would be a great time to come in and talk to me, go over this with me if you're not exactly sure how that works, and we can kind of try and draw it out so you can kind of see it in action. Okay, but hopefully the simulations do some of that for you as well. Okay, guys, that's all I've got for you. I hope you have a great rest of your day. If you need anything, know I'm here. You can reach out through Remind, through Google Classroom private comment, through email, or give me a call, or I'll be there for office hours. All right, have a great rest of your day. Bye.